Good day, this is the Professopher, CBMD PhD, and today's Saturday, December 31st, 2022. It is 4:35 and 32 seconds, a.m. Eastern Standard Time, and today's research, pancreatic cancer. So let's hop to it here. The study discovers triple immunotherapy combination as possible treatment for pancreatic cancer, targeting T cells and myeloid cells together improve anti-tumor response and survival in preclinical models. Date December 30th, 2022. Source: University of Texas. The researchers used comprehensive immunoprofiling in mouse of human pancreatic cancers to systematically identify mechanisms of immunotherapy resistance and investigate potential therapeutic targets. They found that neutralizing several distinctive immunosuppressive mechanisms of the time dramatically improved survival rates in laboratory models, pointing to a potential treatment option for this notoriously lethal and unresponsive cancer. And this triple combination therapy led to an unprecedented curative response in the models. The prevailing view has been that pancreatic cancer is impervious to immunotherapy, but this preclinical study shows that it can be vulnerable to the right combination therapy. Moreover, the presence of these targets in human pancreatic cancer specimens raises the exciting possibility that such therapeutic combinations can one day help our patients. The pancreatic cancer is one of the leading causes of cancer death in the United States, partially because 80% of cases are diagnosed at the advanced stage. Pancreatic cancer is also considered to be non-immunogenic, meaning it is unresponsive to commonly used anti-PD-1 and anti-CTLA-4 immune checkpoint inhibitors. And this is due in part to the immunosuppressive conditions in the time, but the mechanisms behind this resistance are not fully understood. The researchers used high-dimensional immune profiling and single-cell RNA sequencing to study how the time is affected by a variety of immunotherapies. They identified specific immune checkpoint proteins, 4-1BB and LAG, that were highly expressed in exhausted T cells. In testing antibodies targeting these checkpoints, the researchers observed that models treated with a 4-1BB agonist and LAG3 antagonist in combination had slower tumor progression, higher levels of anti-tumor immunity indicators, and significantly improved survival rates compared to treatment with either antibody alone or with other checkpoint inhibitors. Notably, these preclinical studies faithfully mirrored the human data and their lack of efficacy of anti-PD-1 or anti-CTLA-4 therapy. The researchers also confirmed these two therapeutic targets are present in human pancreatic cancer samples, with 81% and 93% of patients analyzed having T cells with 4-1BB and LAG3 expression, respectively. And because this dual therapy combination did not completely eliminate established tumors, the investigators also examined efforts to reprogram the time to further sensitize tumors to immunotherapy. At baseline, the time contained an abundance of myeloid-derived suppressor cells, expressing CXCR2, a protein associated with recruiting immunosuppressive cells. Inhibiting CXCR2 alone decreased myeloid-derived suppressor cell migration and blocked tumor growth, but it was not curative. This prompted the investigators to consider a combination targeting 4-1BB, LAG3, and CXCR2. It was this triple combination that resulted in complete tumor regression and improved overall survival in 90% of preclinical models. In a more stringent laboratory model that develops multiple spontaneously arising tumors with higher treatment resistance, the combination achieved complete tumor regression in over 20% of cases. As these are encouraging results, especially considering selective effective immunotherapy options in pancreatic cancer by targeting multiple synergistic mechanisms that get in the way of the immune response. We can give T cells a fighting chance to attack these tumors. Of course, we still need to see how this combination translates into a safe and effective regimen in the clinic, and we invite the researchers to build upon these results. As researchers, we are optimistic about pancreatic cancers, and hopefully other non-immunogenic cancers can ultimately be rendered vulnerable to combination immunotherapy. The authors point out that these particular immunotherapy agents currently are undergoing clinical trials as monotherapies, suggesting potential opportunities to rapidly translate and triple combination into clinical studies. And this is the Professopher, Professor of Microbiology, Bioinformatics, and Pathophysiology. This video was created for educational purposes only. Hope you enjoyed listening. Until next time, Happy New Year and have an awesome day.